Hello, my name is Michael Heneka. Me and my team are interested in inflammatory mechanisms in various brain disorders, including neurodegenerative diseases. We have been studying the mechanisms that set the scene for inflammation, such as locus coeruleus degeneration, the role of NSAIDs and NSAID-mediated protection in Alzheimer's disease, and ultimately got very interested in innate immune activation through the NAMP3 inflammasome signaling pathway. Inflammation in Alzheimer's disease. What is the cause and what is the meaning, and why this should be a future research focus? So to consider the meaning of an inflammatory response in Alzheimer's disease, we have to go back and think about what the innate immune system, which drives most of this response, has been evolutionarily developed for. And actually, this is the defense of our body to challenges from outside by fungi, by bacteria, and by viruses. So these innate immune cells, which we call myeloid cells and which include microglia cells, which represent the brain's innate immune system, are actually equipped with pattern recognition receptors on their surface, which sense structures that are foreign to them, and then they elicit an inflammatory reaction, which is one part releasing inflammatory mediators, the other part which is a phagocytic clearance response. So it happens to be the fact that some of the bacteria we know very well, including Escherichia coli, Staph aureus, and Salmonella typhimurium, carry beta-sheet structured amyloids, very similar to those as they deposit in the brains of Alzheimer's patients on their surface by nature, which means that if the myeloid cells, by pattern recognition receptor ligation, sense these structures, they respond in the same way because that's their evolutionary program. Now, if microglia cells become activated, they are not only releasing a lot of inflammatory mediators, they also stop doing a lot of beneficial things for our brain, including generation and secretion of trophic factors, synaptic scaling and pruning, which is necessary to keep neuronal networks going. Now, inflammation has two major consequences. One is a functional consequence. We know that the release of these inflammatory mediators, including L1-beta, TNF-alpha, nitric oxide, or other cytokines, is able to functionally impair neuronal network activity. And a prominent example is the suppression of long-term potentiation in the hippocampus. While LTP is not the equivalent to memory, it's an important factor to memory consolidation and long-term storage of information in our brain. So if that's suppressed, we could conclude that the presence of inflammation impairs the memory function. Now, after or in parallel to the functional impairment, there are structural consequences. So inflammation, of course, does not occur alone in the brains of these patients. There is excitotoxic activity as well, and a paper a long time ago by Sandy Hewitt and Moses Chow in Neuron in 1996 nicely showed that inflammation and excitotoxic stimuli act synergistically and induce neuronal death. There is an effect on synaptic structures, which can be engulfed in a complement 3 receptor-dependent mechanism, and those synapses which are decored by C3 are then taken up by microglia. And that's obviously one of the effects which also happens to take place, at least in models of cerebral amyloidosis, which are linked to Alzheimer's disease. Now, there are other mechanisms by which inflammatory reactions can impair neuronal survival, including phagocytosis. So if neurons do not present do not eat me signals on their surface, then the immediate reaction of microglia cells can be to take those neurons up even alive and digest them. Beyond these direct effects on neurons, there's a variety of other factors. So inflammatory molecules in some of the models have been shown to upregulate APP processing, especially by increasing beta secretase 1 levels, so fueling into the pro-amyloidogenic pathway, which would lead to an increase in the concentrations of beta amyloid peptides. At the same time, inflammation is able to impair the phagocytic clearance of A-beta peptides by microglia cells and maybe other pathways as well. An intriguing mechanism by which microglia can actually interfere with beta amyloid deposition is the activation of the NALP3 inflammasome, which leads to microglial pyroptosis and the release of ASC specs. And ASC specs are highly aggregating conglomerates which interact with beta amyloid and actually seed them. Is a fundamental mechanism involved in the seeding of beta amyloid pathology in response to the injection of APP brain lysate injection into the hippocampus. So obviously this inflammatory molecule can change the propensity of beta amyloid to aggregate and form the seeding core of plaque deposits. And that could be replicated in human plaque analysis as well.